Hello everyone, welcome to biology class. In this class we are going to see 11th standard botany chapter 1 living world. In this part of this video we are going to see about bacteria. Already in the previous video we have seen about bacteria, their size, their shape, flagellation and the special staining technique called gram staining. This class we will see some more details about bacteria. So first let's see about the life processes in bacteria. In the life process first we are going to see about respiration. So respiration, it is a process by which the food taken by the organism will be converted into the form of energy. That is called as respiration. In case of bacteria, two types of respiration take place. One is aerobic and anaerobic. Aerobic means takes place in the presence of oxygen. Anaerobic means takes place in the absence of oxygen. First, aerobic respiration. So already in case of respiration, you would have known in case of aerobic respiration, there are 4 stages in aerobic respiration. You would have learnt in your previous classes. So the 4 events in respiration are glycolysis, oxidative decarboxylation of pyruvic acid, Krebs cycle and electron transport chain. So these 4 processes will be taking place during the process of respiration, and when these 4 processes takes place only, the glucose molecule, which is the ultimate simple form of food, will be converted into the form of energy. So this will take place in the last stage, that is the conversion of food into the form of energy called ATP molecule will be taking place in the last stage of respiration, which is called as electron transport chain. So during the electron transport chain, the terminal acceptor of electron molecule will be oxygen. So that's why it is called aerobic respiration, since oxygen is involved in the process of respiration, and this oxygen acts as terminal acceptor. Terminal means the last acceptor in the chain. The last acceptor of oxygen is the last acceptor of electron is oxygen. So these bacteria cannot grow under anaerobic condition, that is in the absence of oxygen. And example is Streptococcus. Next, the term obligate aerobe. Obligate meaning strict. So those organisms that can survive only with the oxygen is referred to as obligate aerobe. An example is Micrococcus species. Next, anaerobic respiration. So certain bacteria, they do not use oxygen. That's why they are referred to as anaerobic. But they also need energy, and that energy is obtained from fermentation reaction. And example for such anaerobic bacteria is Clostridium. Next, facultative anaerobes. What are facultative anaerobes? These are bacteria that can grow either using oxygen. So they can use the oxygen as a terminal acceptor, or they can also live anaerobically, that is using fermentation reaction to obtain the energy. That's why they are called as facultative anaerobes. So for example, for facultative anaerobe is Escherichia coli. And Escherichia coli, when it is found in the site of infection like abdominal abscesses, abdominal abscesses means in case of intestine there will be a form of lesion which is filled with the pus. That is what is called abscess, and it can rapidly consume all available oxygen. So after consuming all the available oxygen, the prevailing condition in that environment changes to anaerobic. So now this bacteria can act as anaerobic environment, and the anaerobic bacteria that are present can grow and cause the disease. Next, capnophilic bacteria. So capnophilic bacteria means the bacteria which require carbon dioxide for their growth. Till now we have seen the oxygen requirement, anaerobe, aerobe, that and all based upon the oxygen requirement. And this capnophilic bacteria is based upon the carbon dioxide requirement. Those bacteria that require carbon dioxide are referred to as capnophilic bacteria. Example, Campylobacter. So that is it with the respiration in bacteria. The next life process in bacteria is nutrition. So on the basis of nutrition, so nutrition is nothing but the mode of taking in of food. So how the bacteria are going to obtain food are referred to as nutrition. So based on the mode of nutrition, the bacteria can be classified into two different kinds, autotrophs and heterotrophs. First, let's see in detail about the autotrophic bacteria. So what is an autotroph? The organisms that can synthesize their own food. And when it is autotrophic bacteria, the bacteria that are able to synthesize its own food are known as autotrophic bacteria. And these autotrophic bacteria can be further subdivided into photoautotrophic bacteria and chemoautotrophic bacteria. So the next slide we can see what is chemoautotrophic bacteria. Now let's see what is photoautotrophic bacteria. The term photo meaning light, autotrophic means they can prepare their own food. So the bacteria which are using sunlight as a source of energy to synthesize the food are referred to as photoautotrophic bacteria. So this photoautotrophic bacteria may be of two different kinds. So in the photoautotrophic itself, two different kinds. One is photolithotroph and the other one is photoorganotrophs. So the term litho refers to the inorganic substance. The organo refers to organic substances. In case of photolithotroph, so already we know they are going to use sunlight as the source of energy. But for the photosynthetic process, which act as hydrogen donor. In case of lithotroph, the inorganic substances act as hydrogen donor. Make it very clear. Autotrophic means synthesizing own food. Photoautotrophic means synthesize their own food with the help of sunlight as the energy. And photolithotroph means they are going to synthesize their own food by using sunlight as the source of energy, and the hydrogen donor is going to be an inorganic substance. So under photolithotroph, you have two different bacteria under photolithotroph. One is green sulfur bacteria, the other one is purple sulfur bacteria. So in case of green sulfur bacteria, the hydrogen donor is H2S. So here you have to write 2 in subscript. So you have to write below H. So H2S, and they possess a pigment called bacterioviridin. So because they are autotrophic bacteria, they should have some pigment to absorb sunlight. So the pigment that is found in case of photolithotroph is bacterioviridin, and the hydrogen donor is hydrogen sulfide H2S. And example for green sulfur bacteria is chlorobium. The next under photolithotroph is purple sulfur bacteria. So in this, the hydrogen donor is thiosulfate and the pigment is bacteriochlorophyll. And the chlorophyll containing chlorosomes are also present. The units where the chlorophyll are found are referred to as chlorosomes. Example, chromatium. So that's it with the photolithotrophs. The next is photoorganotroph. Again, they are going to make food by using sunlight as a source of energy. But the hydrogen donor, as the name suggests, it is going to be an organic substance. It can be either organic acid or alcohol. And example for photoorganotroph is purple non-sulfur bacteria. Example, rhodospirillum. So we have seen nutrition, two different kinds of nutrition, autotrophic, heterotrophic. Under autotrophic itself, we have two different kinds, photoautotrophic and chemoautotrophic. And till now we have seen photoautotrophic bacteria. So under photoautotrophic bacteria, I have two kinds, photolithotroph, photoorganotrophs. So the next we are going to see is under autotrophic bacteria, chemoautotrophic bacteria. As the name suggests, chemo, chemical. So the source of energy, they cannot use the sunlight because they do not have the photosynthetic pigment. So they are going to use chemical substances as source of energy. That chemical substances can be either organic substances or inorganic substances. So based upon whether they are going to use the inorganic substance or organic substances, they are of two different kinds. Chemolithotroph. So again, term litho refers to inorganic compound. So this bacteria, they are going to use the inorganic compounds to release energy. And examples, sulfur bacteria, thiobacillus thiooxidans, iron bacteria, ferrobacillus ferrooxidans, hydrogen bacteria, hydrogenomonas, and nitrifying bacteria, nitrosomonas and nitrobacter. So these are the chemolithotrophic bacteria. Next, chemoorganotroph. That is, they are going to use the organic compounds for the obtaining energy. And examples, methane bacteria, methanococcus, acetic acid bacteria, acetobacter, and lactic acid bacteria, lactobacillus. So these are the chemoorganotrophic bacteria. So the second kind of nutrition, so that's it, we are coming to the end of autotrophic nutrition and bacteria, autotrophic bacteria. Next is heterotrophic bacteria. What are heterotroph? The organisms that are not able to synthesize their own food and they are going to depend upon other organisms for their food are referred to as heterotrophic bacteria. Under heterotrophic bacteria, you have three different kinds of heterotrophic bacteria: parasite, saprophyte, symbiotic. What are meant by parasite? Parasites are the bacteria that live inside the body of another organism called host and they derive their nourishment from the host. They are referred to as parasitic bacteria. Saprophytic bacteria. Example, Bacillus mycoides. So saprophytic bacteria are nothing but they are going to depend on the dead and decaying organic matter for their food. The next category of heterotrophic bacteria is symbiotic bacteria. So what are symbiotic kind of nutrition? Symbiotic nutrition is a nutrition in which the organisms come into a relationship, that is mutual relationship, in which both the organisms are benefited. For example, rhizobium. Rhizobium is the name of the bacteria and this rhizobium is known to live in the root nodule of leguminous crops. So the legume variety of crops, you can find this rhizobium. They are living in the root of the leguminous plant in the form of a nodule. So that's it with the heterotrophic bacteria. What we have learned now, we can see in the form of a flowchart. So nutrition in bacteria, it can be autotrophic bacteria or heterotrophic bacteria. Under autotrophic bacteria, you have photoautotrophic bacteria and chemoautotrophic bacteria. Under photoautotrophic bacteria itself, you have photolithotroph and photoorganotroph. Photolithotroph is nothing but the hydrogen donor is going to be inorganic substance and photoorganotroph is nothing but the organic acid or alcohol act as hydrogen donor. Under chemoautotrophic bacteria, you have chemolithotroph. That is, if it is going to use inorganic compound to release energy, and chemoorganotroph if it is going to use organic compound, oxidized organic compounds to release energy. So that's it with the autotrophic bacteria. Now coming to heterotrophic bacteria, there are three different kinds of heterotrophic bacteria: parasite, saprophyte and symbionts. So that's it with the nutrition in bacteria. So now we have seen two important life processes in bacteria. One is respiration, the another one is nutrition. The next life process we are going to see is reproduction. So how reproduction takes place in bacteria? Reproduction takes place in bacteria either by means of asexual means such as binary fission, conidia and endospore formation. In that, first under asexual reproduction we are going to see binary fission. So out of all the three, the binary fission is the most common one. So what is binary fission? So this process takes place under favorable condition. That is, the condition that are apt for the reproduction, that is referred to as favorable condition. So the nuclear material, first what is going to divide? The nuclear material of the bacteria is going to divide, and after the nuclear material is going to divide, a simple median construction appears. So here you can find the constriction appears in the cell wall and then the constriction deepens and finally it results in two cells which leads to the separation of the two cells. So this process is called binary fission. Fission means to split or cut. Binary means two. So since one bacteria is going to split into two, it is referred to as binary fission. The next kind of asexual reproduction taking place in bacteria is by means of endospore. So under favorable condition, the binary fission is going to take place, and under unfavorable condition, when the conditions are not up to mark, this kind of asexual reproduction will take place in bacteria. Not all the bacteria have the ability to form the endospore. Only very few bacteria have the ability to form the endospore. And examples include Bacillus megaterium, Bacillus ferricus and Clostridium tetani. So what are endospores? So endospores are nothing but thick-walled resting spore. Okay, so the bacteria during the unfavorable condition, it produces a thick wall, and the bacteria lives in the form of a cyst inside the thick wall, and when conditions turn into favorable condition, this bacteria, the endospore, will germinate and form the bacteria. So this is the microscopic observation of the endospore. So the dark part is the endospore in this microscopic picture. Then sexual reproduction. So till now we have seen asexual reproduction in bacteria. Under asexual reproduction, binary fission taking place under favorable condition and endospore formation taking place under unfavorable condition. Next, move on to sexual reproduction in bacteria. So though in case of bacteria, a typical sexual reproduction is not going to take place. What is a typical sexual reproduction? That is formation of gametes and fusion of gametes. So here no such formation and fusion of gametes. However, the sexual reproduction the bacteria undergoes by means of genetic recombination or gene recombination. So this gene recombination may occur in bacteria by means of different methods. They are conjugation, transformation, transduction. The first process under sexual reproduction is conjugation. Lederberg and Tatum demonstrated conjugation in E. coli in the year 1946. So you have to remember the name of the scientist. So what happens in this method? The gene transfer, the method of gene, the transfer, the donor cell gets attached to the recipient cell with the help of pili. So already when you have learnt about the ultrastructure of a bacterial cell, we have learnt about the pili. They are the appendages which are arising from the bacteria. So when the pili, there are different names for the pili, and when the pili is responsible for conjugation, that pili is referred to as sex pili. So here this pili is going to be helpful for conjugation. So this is referred to as sex pili or conjugation pilus. The pilus grows in size and forms a tube-like structure, and that tube is referred to as conjugation tube. So the plasmid of the donor cell. So the bacterial cell which is having the plasmid is referred to as F+, that is fertile. F here refers to fertility factor. When the plasmid is found, that cell is referred to as F+. When it is not found, then it is referred to as F-. So what happens during the conjugation process? So the cell that has got the plasmid DNA and which is going to donate the plasmid DNA are said to be donor cell. On the other hand, which is going to receive is said to be recipient cell. So in this process of conjugation, the plasmid of the donor cell, that is the F+ fertility factor, will undergo respiration, and out of double-stranded plasmid DNA, only one strand of the DNA is transferred to the recipient cell through this conjugation tube. Only one strand will be transferred, and the recipient, that is after one of the strand enters into the recipient will complete the structure of the double-stranded DNA by synthesizing the strand that complements the strand acquired from the donor. So this process is referred to as conjugation. The second kind of sexual reproduction is transformation. Transformation is nothing but the transfer of DNA from one bacterium to another is called transformation. This was demonstrated by a bacteriologist called Frederick Griffith, and this is demonstrated by using a experiment in mice and using a bacteria called Diplococcus pneumoniae. The other name for the bacteria is Streptococcus pneumoniae. So in this Diplococcus pneumoniae bacteria, two strains of the bacteria are present. So one strain has the ability to produce smooth colonies and they are virulent in nature. Virulent is nothing but having the ability to produce disease is referred to as virulent, and those bacteria that are having the virulent property is referred to as S type. And when it is grown in culture, when the bacteria is grown in the artificial medium, these bacteria produce smooth colonies. Colonies is nothing but the growth of bacteria in the artificial medium will be shown in the form of groups, so they are referred to as colonies. And the another strain of bacteria is nothing but they are avirulent strain, that is they produce rough colonies. Okay, so two strains of bacteria are there. One is virulent strain, the other one is avirulent strain. The virulent strain is referred to as S type, whereas the avirulent strain is referred to as R type. So now let's move into the experiment. So when S type of cells, that is when virulent type of cells were injected into the mouse. So this is the virulent type of cell, that is S cells. When these S cells are injected into the mouse died. When R type of cell is injected into the mouse survived. Then what happened? The third step, heat killed S type. Okay, so the S type, which is known to produce the disease to the mice, when it is heat killed, and then after it is heat killed, if it is injected into the mouse survived, okay, did not die. So what have happened? The heat have degenerated the capsule structure of the bacteria, thereby the bacteria are not able to produce the disease and hence the mouse survived. And the last step, he mixed the heat killed S type and R type, and he thought that the mouse will survive, but unfortunately the mouse died. So what have happened? So we know the heat killed strain cannot cause the disease, and even the rough strain, that is R type cell, cannot cause the disease. But when both the heat killed S and the avirulent rough strain are injected together, there is transformation have taken place. So what is transformation? The hereditary material of heat killed S type cell is transferred to the R type cell, and this gene from the S type cell which was transformed to the R type cell have transformed the non-virulent strain into virulent strain, and this phenomenon is called transformation, and this experiment is also referred to as Griffith's transformation experiment, or simply Griffith's experiment. The third kind of sexual reproduction in bacteria is transduction. So this process was discovered by Zinder and Lederberg, and it was discovered in the bacteria called Salmonella typhimurium. So this phage mediated DNA transfer is called as transduction. In transduction, there are two types of transduction. One is generalized transduction, the other one is specialized or restricted transduction. So already you would have familiar with this process. So in case of the lytic and lysogenic cycle, we have seen how the phage is going to enter. So the ability of the bacteriophage to carry genetic material of any region of a bacterial DNA. So any part of the bacterial, any region of the bacterial DNA, if it is transformed, then it is referred to as generalized transduction. whereas in case of specialized or restricted transduction, the phage can carry only the limited or specific region of the bacterial DNA, and since the specific region of the bacterial DNA is transferred from the phage, this kind of transduction is called as specialized or restricted transduction. So that's it with the life processes in bacteria. So under life processes in bacteria, we have seen about the respiration, and then we saw about respiration itself. We have seen about the aerobe and the anaerobe, the facultative anaerobe and capnophilic bacteria. And then we have seen about the nutrition bacteria, autotrophic bacteria and heterotrophic bacteria. And about reproduction in bacteria, we have seen two modes of reproduction. One is asexual mode of reproduction, and under asexual mode of reproduction, we have seen the binary fission and endospore formation. And under sexual reproduction in bacteria, we have seen three different modes. One is conjugation, number two is transformation, and number three is transduction. Next, we are going to see about the economic importance of bacteria. So, what is the economic importance of bacteria? We know bacteria are both beneficial and harmful. So, in the starting of the bacteria chapter itself, we have seen that certain bacteria are very helpful to us. For example, we saw as an example of lactobacillus, which is important for the conversion of milk into curd. And also we have seen about harmful aspect of bacteria, that is the typhoid fever is caused by a bacteria called Salmonella typhi. So we are going to see in detail about the economic importance of bacteria. And first to move about, we will see about the beneficial activities of bacteria. So under beneficial activity of bacteria, the first aspect we are going to discuss about soil fertility. So what is the role of bacteria in making the soil fertile? So first one we are going to see about ammonification. So ammonification, what are all the bacteria involved in this process? Bacillus ramosus and Bacillus mycoides. So these bacteria can convert the dead bodies of plants and animals will be having certain protein. So from the proteins, they can convert those proteins into ammonia, and which can be converted into ammonium salt. So the process of ammonia formation is referred to as ammonification, and the bacteria that are employed in ammonification process is Bacillus ramosus and Bacillus mycoides. The second aspect in case of soil fertility is nitrification, and the bacteria involved in nitrification is Nitrobacter and Nitrosomonas. So these bacteria can convert the ammonium salts into nitrite and nitrate, and the necessity for the conversion of ammonium salt into nitrite and nitrate is these are the usable forms of nitrogen. The third aspect is nitrogen fixation. Certain bacteria are involved in the nitrogen fixation process. They are Acetobacter, Clostridium, Rhizobium, etc. So the bacteria cannot use atmospheric nitrogen directly. So the atmospheric nitrogen has to be converted into usable form, and this process is called nitrogen fixation. The next beneficial activity of bacteria is antibiotics. So certain antibiotics are produced from bacteria. The antibiotic that are obtained from bacteria, the first one is streptomycin. So this streptomycin is obtained from Streptomyces griseus. This antibiotic streptomycin has ability to cure urinary infection, tuberculosis, meningitis and pneumonia. So meningitis, inflammation of meninges of the brain. Next antibiotic is aureomycin. So this antibiotic is obtained from the bacteria Streptomyces aureofaciens, and this antibiotic act as a medicine to treat whooping cough. So children will be affected by this the bacteria called Corynebacterium diphtheriae, which is known to cause this whooping cough, and this aureomycin is used to treat the whooping cough and eye infection. The third antibiotic is chloromycetin, and it is obtained from the bacterium called Streptomyces venezuelae, and it is used to cure typhoid fever. Next, bacitracin. It is obtained from Bacillus licheniformis, and this is used to treat a sexually transmitted disease called syphilis. And next antibiotic is polymyxin. So it is obtained from the bacteria called Bacillus polymyxa, and it can cure some bacterial diseases. So that's it about the antibiotics. The next is industrial uses of bacteria. Certain bacteria are used in the industry. So what are they and what is its use? Lactic acid. The Streptococcus lactis and Lactobacillus bulgaricus have the ability to convert milk, sugar, lactose into lactic acid. And butter, cheese, curd, yogurt, these are all the products. The milk will be converted into butter, cheese, curd and yogurt, and these are the bacteria employed. In case of butter, Streptococcus lactis, and Leuconostoc citroortum, and in case of cheese, Lactobacillus acidophobus and Lactobacillus lactis, curd, Lactobacillus lactis, and yogurt, Lactobacillus bulgaricus. Next is vinegar. Vinegar is referred to as acetic acid, and it is obtained from the bacteria called Acetobacter aceti. So this bacteria oxidizes the ethyl alcohol, which is obtained from molasses. Molasses is nothing but after the sugar is extracted from the sugar cane, the waste is referred to as the molasses, and from the molasses the bacteria oxidizes the ethyl alcohol. From this sugar, it oxidizes the ethyl alcohol by fermentation reaction, leading to the formation of vinegar, that is acetic acid. The next beneficial aspect of bacteria is alcohol and acetone, butyl alcohol and methyl alcohol. It is obtained from the bacterium Clostridium acetobutylicum, and they are again produced by the fermentation activity of bacteria. Next is retting of fiber. So from the fiber yielding plants, the fibers are separated, and this process is called retting, and for this Clostridium bacteria is helpful. Next, certain vitamins are also obtained from the bacteria. Example, you have Escherichia coli and Clostridium acetobutylicum. So these bacteria, first Escherichia coli, it is known to live in the intestine of human beings and is known to produce a large quantity of vitamins such as K and B complex. And Clostridium acetobutylicum, it is known to produce vitamin B2. The next beneficial aspect of bacteria is curing of tea and tobacco. So curing is a process, and due to this process, a special flavor and aroma for the tea is imparted, and the bacteria employed is Mycococcus candescens and Bacillus megaterium. The next, certain bacteria are known to cause some harmful effects. So now let's see the harmful activities of bacteria. First, to go about the plant diseases caused by bacteria. Certain bacteria are known to cause diseases in plants. And examples we have, so in rice plant, Xanthomonas oryzae is the bacteria which is known to cause bacterial blight disease. And in case of apple, Erwinia amylovora, known to cause fire blight. And in carrot, Erwinia keratopora, it is known to cause soft rot in carrot. And in case of citrus plant, Xanthomonas citri is known to cause citrus canker. And in case of cotton plant, Xanthomonas malvacearum, it is known to cause a disease called angular leaf spot. And in case of potato, Clavibacter mycigenensis subspecies Cephidonicus, it is known to cause ring rot disease in potato. And another bacteria called Streptomyces myces Scabies is known to cause a disease called scab in case of potato. So these are the plant diseases caused by bacteria. Even in case of animals also, certain bacteria are known to cause diseases. So the name of the diseases and pathogen are given in the following table. First, sheep, anthrax disease is caused by the bacteria called Bacillus anthracis. And in case of cattle, so cattle cow, Brucella abortus is known to cause brucellosis, and Mycobacterium bovis is known to cause bovine tuberculosis. And in case of Clostridium chanvei, it is known to cause a black leg disease in cattle. Coming to the human diseases caused by bacteria, there are many number of diseases caused by bacteria. A few are listed here. So cholera is a bacterial disease caused by Vibrio cholerae. Typhoid is caused by Salmonella typhi. Tuberculosis is caused by Mycobacterium tuberculosis. Leprosy is caused by Mycobacterium leprae. Pneumonia is caused by Diplococcus pneumoniae. Plague is caused by Yersinia pestis. Diphtheria is caused by Corynebacterium diphtheriae. Tetanus is caused by Clostridium tetani. Food poisoning is caused by Clostridium botulinum, and syphilis is caused by Treponema pallidum. These are few bacterial diseases in humans. Next, some important do you know? So probiotics. So probiotic milk product, toothpaste are available in the market. Lactobacillus bifidobacterium are used to prepare the probiotic yogurt and toothpaste. And another, do you know, the bacteria may form the biofilm. So and it can lead to dental caries. Dental carie means it can lead to the formation of cavity in the teeth, infection, and even urinary tract infection. And Ralstonia synthesize PHB, polybeta hydroxybutyrate. It is a microbial plastic of microbial origin, and it is a biodegradable plastic. Very, very important objective. So biodegradable plastic is polybeta hydroxybutyrate. And the next, do you know, Pseudomonas putida? You would have heard it is referred to as superbug. It is a genetically engineered bacterium, that is genetically modified bacterium, and it has the ability to break down the hydrocarbon. And protein is a single cell protein derived from the bacteria called Methylophilus and Methylotropus, and it is used as a protein supplement. Agrobacterium tumefaciens. So this bacteria is known to cause a disease in plant, and the name of the disease is crown gall disease. And but its inherent tumor inducing principle helps to carry the desired gene. That is, so though the bacteria is known to cause the disease, and when we eliminate the disease causing capability, and in place of that if we integrate any useful gene, this bacteria will be helpful to carry the useful or the desired gene to the plant through genetic engineering. Thermus aquaticus. So the next topic will be learning about certain specific bacteria, archibacteria and all. So one such bacteria is called thermophilic bacteria, which can live under extreme temperature. It produces an enzyme called Taq polymerase, and it is important for a reaction called polymerase chain reaction, PCR technique. So it is a very important technique producing the many copies. Methanobacterium is employed in biogas production, and halobacterium is an extremophilic bacterium. That is, halo means salinity, salt concentration. So this bacteria is known to survive in high salt concentration, and it is exploited. That is, it is being used up because they can produce the beta carotene. So that's it. We are coming to the end of today's session. So we have seen about life processes in bacteria, of which we have seen respiration, nutrition and reproduction, and also we have seen about the beneficial and harmful activities of bacteria. Thank you. Meet you in the next class.